Hello, and very good afternoon to the distinguished guests, my incredible viewers and dear colleagues. We are, facing some technical issues, so it was a delay for 10 minutes. I really apologize for that. So I am Yasha Nirati, presently working as a scientist in Biocon Limited, located in Bangalore, India. So on behalf of Nyberman Bioinformatics, I'm delighted to welcome each and every one of you today's webinar on optimization of molecular docking, focusing on enhancing precision in drug discovery and design efforts. Your active participation underscores our shared commitment to advancing knowledge and fostering collaboration within the scientific community. This webinar aims to provide insights into techniques for identifying decoys, boosting the enrichment of active compounds, and optimizing docking settings, giving you an opportunity to enhance your structural. So before going any further, I'd like to introduce Nyberman Bioinformatics to all of you at first. Nyberman Bioinformatics, founded in 2019, is a globally recognized bioinformatics service firm with a unique mission. Our goal is to create a comprehensive network that brings together academia, industries, students, and researchers worldwide in the expansive fields of bioinformatics, biology, and related disciplines. So this vision took a significant step towards the launch of Nyberman Bioinformatics Europe in 2021, providing exceptional training and services to individuals globally. Our expertise spans a wide range of areas, including omics, structural bioinformatics, computational drug design, molecular modeling, molecular dynamics simulations, cheminformatics, programming, artificial intelligence and machine learning, systems biology, health informatics, and intellectual property rights. So over the years, we have hosted a numerous amount of workshops and skill development programs in various bioinformatics domains, along with over 300 plus webinars, each attracting an average of 500 plus participants from France and beyond. We remain dedicated to fostering collaborations and offering opportunities for project outsourcing, consultation, freelancing, and customized training programs. For more information and contact details, please visit our website www.nyberman.com. So as we move ahead towards our next segment, I'd like to tell you all that Nyberman team has established a €1 webinar club. So this initiative aims to create a globally accessible educational platform that facilitates a beneficial exchange of bioinformatics knowledge between the aspiring learners and seasoned experts. So the club offers fantastic opportunities for both the groups. Students can develop in-demand skills in bioinformatics and related fields, attend talks and webinars on topics ranging from learning bioinformatics pipeline and tools to building careers in these areas. Meanwhile, experts can earn by sharing their knowledge, unleash freelancing opportunities, obtain valuable recommendation letters and certificates, and maybe have a chance to join a Nyberman as a team. So don't hesitate any further. Subscribe now at www.nyberman.com/webinars/1 Now, viewers, there is something important regarding the study materials and recordings. So study materials and recording of this session will be exclusively awarded to the members of Nyberman €1 webinar club. Mahesh, can you please change the slide? Dear audience, there is some technical issue. Maybe they are not able to. I hope I'm audible to everyone.
Yes, Asha, you're audible.
Okay. Thank you so much for the confirmation.
You please go ahead with what you want to speak, please.
All right then. Okay. So, yeah, we were talking about the recordings and the study material. So, in case you need the recordings and the study material of the session, it will be exclusively awarded to the members of the Nyberman 1 Euro webinar club. So, if you're already a member of the club, then a feedback link will be emailed to you once the webinar ends. But if not, then there's nothing to panic. You can subscribe now at www.nyberman.com/webinars/1 Euro club. I would request the team that could you please paste the link once in a chat box so that people can go to the link immediately. Okay, so now we come to the certification process. So, dear attendees, to ensure that only those who have participated receive the certificates, we kindly ask you to follow two basic steps. All right. So, the first is to request the feedback link from our Instagram team via direct message, and then complete the feedback form to receive the certificate via email. So the important thing here is that we send you an attendance code in the chat box, both on Zoom and on YouTube at any time point during the webinar. This code will enable you to request the feedback link and fill out the form. While submitting it, again, you'll be asked the same code. So do check on the chat box and note the attendance code. Through that, you can avail your certificates. So stay alert. Now, it's time to welcome our guests for today's webinar. So we welcome Mr. Rick Ganguly. He's currently a junior scientist at Cellogen Therapeutics, while also pursuing research at Northeastern University, specializing in computational biology and bioinformatics. With a solid foundation in mathematics and biotechnology, its expertise propels innovative projects aimed at developing advanced therapeutic solutions. Throughout his academic journey, he has gained numerous biotech skills and consistently shown a commitment to advancing knowledge and research in molecular docking, molecular dynamics, cheminformatics, and Python programming. He works alongside a team of dedicated scientists to explore new avenues in computational biology, contributing to the design and execution of experiments, data analysis, and result interpretation. So we are honored to have Mr. Rick join us as a guest speaker at Nyberman Bioinformatics Europe. So before handing it over to him, I would request all the audience to please collect their questions. We'll definitely have a Q&A session followed by Rick's presentation. There you can ask your questions. So, Mr. Ganguly, once again, we welcome you for today's webinar. It's your time now. Please go ahead with your presentation. Mr. Ganguly, are you there?
Mahesh, can you please make Ganguly as a host?
Hello, am I audible?
Yes,
Hello.
sir, you're audible.
Yes, you are. You are.
Okay. First of all, I'm really thankful to the Neighborman team and everyone for inviting me to give a lecture on molecular docking and optimization of docking protocols. So should I share my screen or the slides like here?
Yeah, sure, please.
I'll go ahead with the slide. Share. So I hope it is visible.
Yeah.
Okay, so first of all, I want to talk about why we need to study this molecular interaction optimization of molecular docking protocols. So molecular docking and in silico molecular interaction studies is a widely used in silico studies that nowadays a lot of people are doing. And in a lot of different type of research, we use like molecular interaction studies where we get a computational microscopy, which normal wet lab we cannot achieve. So doing a molecular interaction studies is important, as well as optimizing a molecular interaction studies is very important. So in molecular interaction studies, there are various types of interaction that we can study. So mostly we study protein-protein interaction and protein small molecule interactions. So we call the small molecule as ligands. Generally, we call it as ligands, and we need to predict if the ligand is binding to the active pocket precisely, and it is working like it should work when we perform a wet lab experiment. So predicting a molecular interaction needs a particular search algorithm. And we know there are n number of molecular docking protocols and softwares and tools that are available. There are several servers which are dedicated for those type of studies. So at least 10 different types of studies that can be done with a search algorithm includes rigid docking, flexible docking, systematic docking, stochastic, deterministic, hybrid approach. So all these different types of docking protocols are available, which has various types of, again, various types of scoring functions associated to the search algorithm. So our goal should not be just to get a score while we are performing a protein small molecular interaction, but also to look into the diverse specification that a molecular docking should need. And we should actually focus on the precise sensitivity and accuracy of the scoring protocol. So various factors such as active site shape, amino acid composition, which forms the active pocket, and even the structural flexibility that can actually give us a various types of diverse result. So coming into the molecular decoys. So what are molecular decoys? So molecular decoys are basically compounds which are structurally similar to the biologically active molecules. Now they might not be interacting equally well when we generally do a molecular inhibition or a enzymatic inhibition of a protein with a small molecule, a designed small molecule. So many a times, irrespective of getting a very good molecular interaction score, many of the compound fails in the wet lab. So it is very necessary to remove the false positive hit from your docking interactions. So today's webinar, I will be focusing mainly on how you can optimize your docking protocol so that you can get a better result in wet lab. So there are certain, sorry for the change. Okay, there are certain things that we have to consider for that. So the most important thing is validation of the docking algorithm. So decoys are used to validate and benchmark the docking algorithm by comparing how well a docking algorithm can differentiate between a true ligand and a decoy. So to do that, we have to understand how a molecule interacts with a protein, and also we have to understand how we can minimize the false positive hits from a docking program or a docking algorithm. That can improve not only our scoring functionality, but also it can reduce the false positive hits. So these are the majorly used servers or tools that we can use to find those docking, those decoy sets. So molecular decoys can be like available by using the DOOT-E server, which is majorly used. And also there are some GitHub tools such as Decoy Finder and many others. But mostly this DOOT-E server or Decoy Finder, they focus on ligand-based approach. Like DOOT-E server have dedicated 102 available protein targets. But looking at the protein universe, where there are like several like lakhs of different types of protein of diverse family and domain, 102 protein is a very small set where you may not find your target protein within this decoy server. So you have to generate a decoy based on their molecular properties, or we can also say the pharmacokinetic or the pharmacophoric properties of a ligand. So where they use a decoy, where the decoys are mainly found based on their structural abilities of the protein to bind to that target, and they have dedicated server for that. Decoy Finder is another such tool that you can find in GitHub, but they all mostly focus on the ligand-based approach than the structure-based approach. So this work of decoy finding, or the interest that I had in decoy finding, started in 2022. So we were working on a paper that is the discovery of a novel inhibitor against urocanistide plasminogen activator. So while working for this paper, we have got certain reviews from the reviewer, very insightful reviews where the reviewer has actually asked for the decoys. So what about the decoys? Like, yeah, you might get a very good score while doing your molecular interaction studies. Everything looks very fair in your paper, on your manuscript, but what about the decoys? Have you considered the false positive hits? So then we started looking at different available tools and servers that are available for looking for decoys. But unfortunately, not a single server had a really good decoy collection of decoy set. So then we thought, what if we can actually exploit certain databases which contains a large number of information regarding the available ligand based on literature and the experimental data. And we have considered using the Campbell database, which is a diverse data set which contains extensive bioactivity data of several different wide range of compounds, and which is also reliable and which can be curated for scientific literature and as well as for molecular interaction studies. So the scoring function limitations includes capturing the binding affinity in a very approximately faulty conformation. So in a major docking drawback is that the ligand become flexible and the active pocket become rigid. So in a rigid docking, most of the general algorithm that we use, whether it is autodock or autodock VNR, or it can be gold. So these type of protocols have a limitation where the ligand is flexible and it can change the conformation based on the rotational liberty or the stereochemical property of the active pocket, where it can rearrange itself to a degree of iterations. So the conformation flexibility also matters. Even the planarity of the molecule also matters. So considering all these things, we had to dive in deep into the molecular properties of several different known inhibitors. So knowing your literature is very important. First of all, to do decoy study, you have to understand that is there any available resources which tells about the known inhibitor and their chemical properties. So to do the enrichment analysis or to compare the molecular interaction protocol, there should be a statistical basis to do so. So in today's world, a lot of different protocols are being verified by this ROC AUC curve. So which is a receiver operator characteristics curve. So what is basically a receiver operating characteristics curve? It gives you a statistical score which tells like the specificity of that protocol versus 1 minus the sensitivity of that protocol. And it also tells, you can see the area under the curve based on your parameters of choice that you use. Here we use the scoring for the scoring scores, I mean the scores of the molecular interaction. And we have compared different types of protocols for different types of docking protocols, as well as you can also check the performance of different type of scoring functions. Like Gold comes up with 4 different scoring function where a lot of people might have a confusion. Shall we use the Gold score or a chem score or some other score that scoring function that are available? And we know that each protein is dynamically different and active pockets are also varying. So deciding on a particular scoring function out of randomness might create a lot of false positive hits. So ROC AUC curve can come into a picture where you can actually define that which of the scoring function is performing better than the other scoring functions. So benchmarking and refinement of your docking protocol is a very important step before starting any in silico molecular interaction studies. So this was the basic plan of work that we have followed, where we have used Campbell Data Client, which is actually a programmable tool that Campbell has provided, where you can use this Campbell Data Client in any Jupyter notebook or IPython notebook, and you can collect n number of different types of characteristics regarding the small molecules that are present there, based on your specific to your target. So these are based on the literature and the experimental data that Campbell has produced over the year, and you can curate those data and convert it into data sheet or data frames using pandas. So with the emerging techniques and technology in bioinformatics, there are a lot of different studies people are doing, and there are millions of compounds that are available, but it's like finding a needle in the haystack, where you have to find one perfect compound and a perfect protocol that actually helps you to define a very perfect score that is not a false positive hit. So looking at 2.5 million chemical compounds that are available in Campbell database, using the in-house script that I will show later in the hands-on session, you can divide the available data for the small molecule associated to your protein target into three different groups. That is inactive, or which are decoys, and active molecule, which are like having a interaction or a binding affinity of like less than 1000 micromolar, and rest all will fit into the intermediate group. So from there, you can have a particular set of compounds which are decoys, and you can carry forward some molecular interaction and get a score of those decoys. And based on like different scoring algorithm, you can or a scoring function, you can check the difference in the scoring matrix, and then you can compare based on the AUC and ROC curve, where the ROC will give a score where receiver operator curve to a threshold of 0.5 is basically will give you a random selection. Anything near from 0 to 1, where any score which is coming into ROC more than 0.5 is more specific. So you also not only have to optimize the scoring function, but also the threshold of the ROC curve. So these are two major challenges to define a very near perfect scoring protocol. So this is a very basic methodology, uh, which is oversimplified for a wide range of audience. So first of all, we have to select the target specific bioactivity data from Campbell using the Campbell data client. This will be specific to the protein target that you are focusing on. And then we have to do a lot of data pre-processing. Why do we need to do data pre-processing? Because a lot of chemical compound comes with certain data where there are so many missing data. Missing data as in like they might not produce some bioactivity score, or like they might not have certain smiles associated to the compound. So all those data has to be dropped, and then the final data that you have in a new data frame, you can do a dimensionality reduction. Now, dimensionality reduction means like there can be n number of features that are associated to a particular compound, and you have to focus on particular feature that you need to check for your interactions. So I will come into all these things. I will define into more finer details when I actually shift to the hands-on session. So, and selecting threshold, like I already mentioned, is a very important characteristics, where here we can see that we have kept the threshold of less than 1000 micromolar for active compounds, and more than 10,000 as inactive or decoys, and anything which fits in between, that is coded by the else function, else loop, it goes into the intermediate data set. Then you can carry forward this decoy molecule and active molecule into molecular docking protocol. that you want to compare with, where you can get a particular score which will fit into the ROC curve. And whichever give a higher area under the curve, here we can see that we have used 4 different docking presets. These are all gold preset, where gold comes with 4 different presets like gold score, chem score, PLP, and ASP. So where gold score have outperformed all other scoring protocols. Here, data normalization is also a very important factor, because not every score which is like giving a higher score is performing well. So some scoring function might have a like lower score is better. So there you have to do some data normalization. So after normalization, you also need to validate. Like it's not that we got a good set, a good protocol that's showing a good ROS area under the curve, and we can carry forward with that, saying that this is the best interaction that we can have with this protocol. So validation include, like, you have to cross check with your active molecule to decoy set and check if the active molecule is having a higher score using that preset than your decoy. So this type of comparison or validation is required. And you also should have an intuition where you can change the threshold of the particular ACRC curve. So all this work I have, this work was inspired by Shenin, and he's also known as data professor. So he have a dedicated YouTube channel also, like, and we I have learned many techniques, like where he have taught like regarding how to get a bioactivity data of from Campbell databases, and I worked on it and modified specific decoy set based on those learning. And I'm also thankful to various resources that I have used, and so that they have made these resources free for research purposes. And I also acknowledge like Northeastern Hill University, where I'm working, and also Cellogen Therapeutics for providing the computational resources where I'm working right now, and many others actually. So now I can move to the hands-on session. So before moving to the hands-on session, I can actually take certain queries. If someone didn't understood anything, I can actually have a like session for that. Like I have time for that as well.
Mr. Ganguly, we have a Q&A session once we get complete the time. Then we
Uh,
can questions.
okay, give me a minute. Actually, I can stop sharing this one. So should I go ahead with the hands-on session, or should I wait for the questions?
Is anybody having any question? They can, I guess, unmute themselves and ask. We can have a small 5 minutes of Q&A session regarding the theoretical part. Okay, I don't think anybody is having
Go
anything.
ahead.
You can go ahead.
Okay, so this is a typical Google Collaboratory notebook where I can uh, show like how we have actually curated the biologically active molecule as well as the decoy set. So looking previously, I have discussed about the problem with different types of pre-existing decoy servers, where you either can go for a ligand-based approach and generate a new decoy dataset from there, or you can look into the available structure-based data that they have provided. So based on those limitation, like Campbell database and the Campbell data client has come into picture, and it has helped tremendously in searching for very specific decoys for a particular protein, that protein of interest. So here you can use the same collab notebook for looking for the decoy dataset for your protein of interest as well. You just have to tweak few of the keywords and few of the things that I will show. So this tells about the basic information about this notebook, what it can do. and I have optimized it for gold. You can use any other protocol. You can also check different types of docking algorithm that are available, and you can compare. For example, you can compare like if autodock or gold, like which one is a better scoring algorithm for your particular protein of interest. Though both are genetic algorithm, but there are slight differences. You will not get the same score in autodock as when you run it through gold, because gold has different type of scoring function. Um, so
Sir,
yes,
sir, how can we? Is it comparable to Schrodinger's accuracy?
uh, Schrodinger also have Schrodinger is very precise. Mainly, Schrodinger doesn't show a lot of false positive hit, as per my knowledge. But they already have optimized. But Schrodinger is also like a paid software. A lot of computational biologists, they mainly focus on the freely available software that are available, like Gold and Autodoc and other resources that we have. So mainly this, I wanted to show how you can compare that what is the best fitting protocol for your protein of interest.
Yes, sir. Thank you.
So,
Yes.
sir, how we will decide whether we should go for structural based database or ligand based database for decoy?
Uh, See, first of all, structure-based database and ligand-based databases are like Dude eServer. I said it has both. It has data for like that 112 protein structures that they have mentioned, or they have like you if you provide your uh, known inhibitor or known ligand, it can generate like decoys on the basis of those information actually. So you can go for go both ways actually. But this one, by using this tool or this process, you can actually generate, you can curate the data from Campbell database, the data associated to your protein. So you can use any protein actually. You don't have limitation to use like those 112 or like any a particular fixed set of protein information. Like for example, galactine is a protein. Very less information is available. So for galactine, specifically galactine 3, you can actually use this database to actually create use this Google Colab notebook to create your set of decoy data, and then you can go ahead with molecular interaction and even molecular dynamics.
Okay, sir. Thank you.
So I think I should go ahead with the rest. So first of all, this is a program that you can see pip install Campbell Web Source Client. So this is the one that I was telling, Campbell Data Client. So we need this to actually curate all the Campbell database data. So we can install this by just simply typing pip install Campbell Web Source Client with the exclamation. I'm using exclamation because I'm in a Google Collaboratory notebook. You can avoid this exclamation if you are running it in local Jupyter notebook actually. So after that, we need to import certain libraries. These are the important libraries for handling the missing data and like forming different data frames to curate a new data from a pre-existing data. So all these things are there. Like Pandas is a very useful library where you can do a lot of data manipulations and data handling. So coming to the step two is search for the protein target. So here, this is the game changer, which I was telling. So we have worked on Galactin 3. Recently, we have communicated one paper based on Galactin 3. So I will keep my discussion limited to the decoy set only. So Galactin 3 also, like I will just tell a little bit about this work. Like Galactin 3 also has an intrinsically disordered region in the protein. So it is a very unique type of protein, have a very less information. So like we thought of trying to find decoy data set for Galactin 3 as well. So in this particular section where you can see, we are actually mentioning our target. Here you can see I have mentioned Galactin 3 as a keyword. So you can replace this Galactin 3 with any of your protein of interest. And if there is any available data or experimental data available in Campbell, it will pop up. So how it will pop up, it will look like this. This is the result section of the page, where you can see like there will be a target chemical ID with a particular number denoting the particular column, and with a preferred name, that is galectin-3 organism and target type. So we wanted to look into Homo sapiens, or the galectin-3 from Homo sapiens as an organism source. So we can select this particular index, that is 1. So in the next session, we have to tell the identifier or the index. So we selected 1 here, as this is coming. This will select this particular galactin entry from Campbell database, which is having the organism information from Homo sapiens. So it will also provide the relevant Campbell ID associated to that particular index. Now, after that, we need to retrieve the bioactivity data for MTS4. That is the whole purpose that why we are doing this particular data analysis. So we converted this particular galectin-3 Campbell database 2318 into a data frame, where you can see it's a huge table, like it's a long list of information that you can get like out of it. So there are, if you go down, you can see there are 95 rows and 46 columns. Like that's a lot of information. So out of this, all this information, the main thing that we are going to do, we are going to curate only those information that are necessary for our study. So we have to go into data handling. So first thing in data handling is handling the missing data. So missing data basically includes those standard values, which tells us about like if there is any inhibitor which can inhibit the protein in the scale of like micromolar to nanomolar concentration. So if it is missing, then it is of no use. And if the canonical smile of that particular compound associated to that particular protein, then also it is of no use, because in the end, we will see that we'll get these canonical smiles associated to the particular compound. So canonical smiles are basically simple representation of the ligand. So we get only the standard values, and not NA is a function that is to drop the missing data. So it will drop all the missing data, and it will give only those entries which have a complete data set. Uh, Hello, can you please mute?
Yeah, we have. You can go ahead.
So after this, we can see there are like total length of this table is 93. So there are 93 different entries within the table. And now moving forward to like the next data pre-processing stage, where we have to check only for a particular dimensionality reduction of the this huge table into our table of interest. So our table of interest include mainly the molecular or the Campbell IDs of those molecules, and the canonical smiles associated to those IDs, and also the standard values. So these are all the bioactivity classes that we have considered, where we have taken only the selection parameter as the molecular Campbell ID, canonical smiles, and the standard values associated to those compounds. So it has reduced that huge table into a very small table with 3 entries, including these 3 particular information. So we can save this as a CSV file. So this program, this particular string of code is doing the same. It is saving this refined table into a data frame 3, that is DF3 to CSV. And we can name it as anything, like I have named it as pre-processed bioactivity data for galectin-3. You can name it anything as per your own will. So the next step is labeling those compounds as either being active, intermediate, or inactive molecules, or molecular decoys. So our rationale was to actually, it is not like you can actually have to go through the literature to know like what are like the threshold that you can set for your particular protein of interest. So 1,000 micromolar is a very huge range to select as active compound, but we didn't want to miss any of the compounds, so we have selected a wide range of compound within the 1000 micromolar concentration. So and we have considered them as active. You can fine tune it to like 100 micromolar or any preferred level. Like you can actually change this level to like select like what should be your active data set and what should be your decoy data set. So decoy data set, we have kept like 10,000 micromolar or more. So anything that comes beyond that will be considered as a decoy. So all the experimental data that are stored in Chemical, it will collect only those as decoy which has particular bioactivity data showing more than 10,000 micromolar concentration. So from there, we have actually can see the next table has another class which shows inactive or decoy molecules and also active. So it is showing only a few entries, like there are 92 entries, among which you can see like a lot of entries are missing here. It's only showing the highlights of the table. So to visualize the number of active, inactive, and decoy, we had to draw a particular graph for that. So we have used Seaborn to visualize this all these molecular characteristics of the different data sets, like whether it is falling into like active, inactive, or decoy. So this is the particular frequency graph that we have generated, where you can see that almost more than 60 molecules are showing like active characteristics of an active compound, and almost 35 molecules have shown to have characteristics of decoy compound. So now the final challenge was to convert it into a molecular smile. So why do we need this molecular smile? Because this is a simple representation of any of the small molecule. So we can totally save this. So this is a print function we can use to select those only the B, table B, that is like the smiles. So we have selected only the smiles, and we can save it in a Word file or save it in a text file. And that text file, we can actually save it with an extension of SMI that will represent the smiles. So, and push it to OpenBabel or any other software which can convert this data into a molecular structural data. So here I have even passed on the code, like you can totally use OpenBabel and pass this like this data set of smile, and you can get an output file in SDF format or MOL2 format, whichever is the preferable format for you to dock or use it as a ligand for docking experiments. And the Gen 3D function in OpenBabel can take care of like the 2D molecular smile and generate three-dimensional structure out of it. So this you can take it further by optimizing the structures or minimize the structure using Avogadro or any other like any other tool that you can use to minimize the structure in a proper order, where there will be no steric hindrance or clashes or contacts. And you can also use this as a save the whole thing as a single PDF or SDF file by using this command in OpenBabel. So this is the part where actually I have discussed only the molecular decoy identification, and as well as like the active versus decoy identification. And we can actually, after this, the molecular interaction, that experiment that you're doing, it might vary. So several use case scenarios, suppose you are using this particular algorithm to check for the best fitting docking scoring function. So then you can choose like a particular scoring function and you can compare with another scoring function, or you can use different algorithms and you can check based on those algorithms. So you can plan accordingly, like what type of docking experiment that you can do out of it. So after that, the ROC and AUC curve, you can plot in any of the available software like SPSS, or you can even use Python notebook. Like I also have a Python notebook. I will just share.
Mr. Ganguly, are you sharing your screen right now?
Yeah. No, I am not sharing. I will be sharing.
Okay.
Give me a minute. Yeah, is it visible?
Yes, yes, yes.
So this is a particular, is it visible?
Yeah,
Hello.
yeah, it is, it is.
Okay, so this is the final notebook which can be used. Like this is a particular example set I'm showing how you can plot a ROC curve or AUC curve. So
Uh,
it's
Hello. Actually, we can see a screen having credits and acknowledgement.
no, actually, I think wait, I'll start sharing again. Give me a minute.
Yeah.
Okay, I think it is visible now.
Yes. Yes. It is showing the Google Notebook.
Yes. So this is a typical example of a Python notebook command. Uh, okay, if you are not comfortable or any of you are not comfortable with Python, you can also use like pre-existing tools such as SPSS. There also you can plot a nice AUC curve, or it comes very handy when you use Python programming for this type of data analysis. So this is just an example I'm showing. Like after you get the decoy molecule, active molecules, and all those data, how you can plot an AUC or ROC curve. So this is the last stage of the analysis where you can actually differentiate between the different scoring function or the different docking algorithm based on the performance, how they are showing in AUC and ROC curve. So here you can see like you have to load the data of your molecular interaction in a particular data frame, and you can populate a nice graph like this type of a graph you can see where there is only two different scoring function of gold, like where there is one showing gold score, another one showing chem score, where you can see the area under the gold score is quite in the positive side as compared to the chem score. So from this graph, you can totally see for the protein of interest that you have used the docking scores, which include all the binary classification for your positive set and also your decoy set. You can totally say that for your protein, the gold score might perform better as compared to the chem score. So by this, it is the end of the session, and I am totally free now for any questions you can ask.
So, dear audience, we have opened the Q&A session time. So you can unmute yourself and ask questions.
So Google Colab notebook that you showed.
I'm sorry, hello, you're not audible. Can you be a little louder?
I was asking if the Google Colab notebook you showed earlier, which was the decoy finder notebook, is it publicly available?
Suzanne, we cannot hear you.
What I understood, like I think Suzanne is asking if it is publicly available, right, Suzanne?
Yes, that's correct.
Yeah, actually, this is a compilation of a lot of different resources that I have read. I have uh, used my, I took help from my lab mates also, and we have developed this code partially. Partially, we have got from, we have collected some data from Shinan's work, and also like we have compiled this code for our purpose. So it is not publicly available right now, but I can make it available in GitHub any soon. Like as per the, if you need, you can contact me also. I can help you out with that.
Anybody else? We have limited time. You can unmute yourselves and ask.
So,
Yes.
earlier you told that ROC value greater than 0.5 will be more specific. So, sir, how you are deciding this value to be
Okay,
specific?
you're talking about the cutoff, like the threshold, right?
Yes, sir.
Yes, see, it depends on your data set also. Like, for example, you have to understand like at which threshold you're getting less false positive. So for that, that is advanced level work where you can actually have to go through some confusion matrix that you can populate. So due to the limitation of time, I could not show a lot of different analysis that falls in between the ROC and finding the decoy set. So a simple way to do it also, like you can visibly compare like the known compound and the decoy set. If you dock it, you will get to see that there are certain scoring functions which are showing higher score for the decoy molecules. So that means that algorithm is unable to determine the active versus decoy. So I hope I'm clear, or should I simplify it more?
So, so please clarify.
Yeah, okay. For example, like if you have like 10 compounds, and out of which 3 are reported drugs for the compound, and 8 are like the decoy, or 7 are like decoy. So now those 3 known compounds should have a higher score, because those are like the known inhibitor which should give a higher score. And those decoys are the molecule which are good binders but are not the inhibitors of that enzyme. Those are like false positive hits. So technically, those false positive hit, if your algorithm can determine that, okay, your particular known compound is giving a higher score, comparatively higher score than decoy. So you have to attenuate the threshold to that level that where it is fitting into like your observed desired result actually. I hope I'm clear.
Yes, sir.
And 0.5 is basically where you are keeping it a random selection, where the score varies from 0 to 1, and anything above 0.5 is being more specific actually. So it depends on your ROC score, like what ROC score also you're getting. It can go below 0.5 as well, but it should be comparably higher than the other algorithm.
So one more question I have. So we have different scoring method like gold score, K score, etc. So how we are deciding which scoring method we should use or which scoring method we should consider?
Yes. So if you are using gold, then you know there are 4 different scoring functions, right? So out of those 4 different scoring function, actually, you can run like parallelly, like see, ROC is basically a binary classifier where you can mention particular active molecular standard known inhibitors as one, and within a particular data set, and you can denote like the decoys as zeros, and you can put it through a ROC curve, and you can see like if the area under the curve for whichever scoring protocol, like scoring function, is giving a more area under the curve. So if your goal score is giving like more area under the curve value, then I think you should go for the goal score. Or if your chem score is giving more, then you have to go for the chem score.
Okay, sir. Thank you.
And there is a lot of data normalization also takes place, because suppose there are certain scoring function where the lower score is better as compared to the higher score. There you have to normalize the data. So there the difficulty increases actually.
Yes, sir.
Any more questions?
Sir,
Hello.
another general question that I have is that you said we could create decoys using databases for the ligand, right? So why can't we do the same for the protein pocket? Because that would become more easier compared to for the ligand. That's what I feel, but I want to just know.
Yeah, it can totally be it can be doable. Like that's what 2D server is doing. 2D server, they have curated the protein pocket, but looking at the diverse like protein that are available. Like if you go to PDB, there are millions of protein that are available. So now you have to use either generative AI or like some other type of um, programming that can actually take care of like that where that can be trained. So it is doable actually. What you're saying, maybe in near future, or maybe a lot of people are doing actually. You can check into that.
Okay, sir.
Hello, sir. I just want to know what are the force field is used in gold as compared to in other. Is there any?
Hello.
Hello,
Hello, Meenakshi.
Yeah,
I did not
Meenakshi, we
get
lost
your
your
question.
voice.
sir. Just want to know the what are the force field are used in gold?
Okay. Gold is basically a docking algorithm. So type of force field, I am actually like right now I cannot recollect the type of force field it uses, but force field we generally use for molecular dynamics simulations. So it has a lot of scoring. Uh, the scoring is based on different type of parameter. So like there is a rotational value, there is a value for hydrophobic interaction, hydrogen bond interaction, and different types of constraint and restraints are there, like that gold has in its formula. So if you go through the gold manual, you will be having a very clear idea about what I'm trying to say and what you want to know. Like you can actually find it in gold manual.
Okay, okay. Thank you.
Okay, we'll have one last question now.
Hello.
Yep.
Hello, Darshan.
Sir, you are suggesting something instead of this Python book. You are suggesting certain alternative. What is it? What was it you're
Okay,
telling about?
alternative could be like a bit tricky, and it might need a lot of computational resources as well as programming knowledge, where you can select like a data set, a curated data set of proteins and their active pockets and the co-crystal ligand. So if you have like the types of interaction and the number of interaction, that can be reduced down into, there is a very good example I will tell you. There is a tool called Dragonfly. It is a recently published tool, like where they are studying like the molecular interaction as interactome, and those interactome are converted into graph network, like where nodes and like edges are being used. So that reduce the dimensionality and the computational timing. So that way people are actually working on those type of things. If you go to the documentation of Dragonfly, you will know what I'm talking about.
Okay, sir. Thank you.
All right then, we'll be closing the Q&A session now. Okay, so Mr. Ganguly, thank you so much for
Thank you
your
so much.
presentation. All right, maybe we Nagalakshmi has some questions. Nagalakshmi, actually we are not able to hear you. Are you trying to say something? Hello? Hello, Nagalakshmi, can you hear me?
Hello.
Okay, we are
Hello.
not able to hear you. Yes, Shiva. Hello?
Are you audible? I have one doubt, ma'am.
Yeah, please ask. This is the final question we are taking. Please go ahead, Shiva.
Hello, sir. Are you audible? Audible voice?
Yes, Shiva, you're audible. Yes.
Is molecular docking autodock variance gives a result as accurate or approximate?
Sorry, I didn't get your question. Can you please repeat?
Sir, for molecular docking, autodock variance software will give a accurate result or approximate.
Um, can you repeat your question? I'm so sorry.
Shiva, can you please speak a bit louder?
Hello.
Yes, Shiva.
Sir, if autonomous software for use to docking, is this result is more accurate or approximate?
Can the host like let me know what is the question? I am unable to understand. I'm sorry.
Okay, sir, no problem.
Shiva, you can actually contact me. I can clarify after the session also if you have any doubt or
Yeah,
any
sir,
of.
sure, sir, sure, sir, sure.
All right, so we wind up now. Hello.
Hello.
Yeah, can you hear me?
Yes, you're audible. Yes.
Yeah, okay. So thank you so much, Mr. Ganguly, for your presentation. It was outstanding, seamlessly integrating diagrams and photos to create an engaging visual experience. Your thorough exploration actually of the concept was remarkable. Starting with the purpose of docking optimization and then introducing the basics of decoys, then building on that with a detailed explanation of the methodology, followed by hands-on demonstration. So we are very thankful to you. The significance and the relevance of molecular docking in today's landscape is undeniable, and your session has given our viewers a fresh perspective and deeper insights into this trending topic. So thank you very much for your time, for your efforts. Thanks a lot. Now, dear audience, I'd like to repeat once again that the study materials and the recording sessions will be exclusively awarded to the members of Nyberman 1 Euro Webinar Club. So please subscribe now at www.nyberman.com/webinars/1 Euro Club to become a member. Please ensure that you note the attendance code, which will help you to receive the feedback link and get the certificate, and enter your name and email address correctly. So feel free to share any concern regarding the tutor or the organizers, as well as suggestions for future sessions through the feedback link that we'll be providing through the email. So moreover, we kindly request that you share your certificates on your social media platforms, tag our account, and help us to spread our word about our upcoming events. To stay updated and learn more about bioinformatics, you can visit our Telegram channel as well, named as Bioinformatics Forum. So before leaving, I want to once again express my heartfelt gratitude to each and every one of you here. We look forward for active participation in our future webinars. Until then, take care. Bye-bye. Thank you so much. Thank you, everyone.
Thank you.
Thank you, Isha.